Okay, welcome to the review video for genetic engineering. In this video, we're going to review three topics that we talked about in classes last unit: transformation, gel electrophoresis, and cloning. So that's what we'll get into right now. Don't forget that genetic engineering is the direct manipulation or changing of an organism's DNA. We can do that in a few different ways. And create organisms out of it that we want, which are genetically modified. So that's what we're going to start with here. How do scientists get this done? How do scientists create organisms with extra traits or extra genes? Well, it starts out with a few elements, okay, a few pieces. The first piece is our plasmid. And the plasmid in this example is a circular piece of DNA. These are only found in bacteria, and they are used very easily in the transformation process. The next element is actually not on here, which is the gene of interest. Okay, I'm going to add that in here. Gene of interest. And that's the gene you're looking to add to a particular organism, and you would combine that with the plasmid. Finally, the restriction enzyme is the last element used in this process, and the restriction enzyme has the ability to cut the DNA in a specific spot. What that allows the scientist to do is to create a plasmid with a particular section that is the gene of interest. And in that way, he has combined, or she has combined, two different pieces of DNA into the same strand. Okay, so how does that get done? Well, you first got to take the plasmid out of the bacterial cell. After taking the plasmid out of the bacterial cell, you cut it with restriction enzymes at a particular location. You use the same restriction enzyme to cut both the plasmid and the DNA, and then you insert the new DNA into the plasmid, and you have your piece of recombinant DNA. Okay, so after this process is over, you've made recombinant DNA. Following that, all you have to do now is place it in a bacterial cell, and as they reproduce, they create clones of the gene you want, okay? That gene of interest that you originally copied. The new DNA in the bacterial cell, again, is called recombinant DNA or combination DNA, okay? We're combining a plasmid with another gene, and that can give us many different combinations depending on what gene you're putting in there, as many as you could possibly think of. You can put a lot of different genes into a plasmid. Here's a pictorial diagram of how it's going to work. Here's our vector. Our vector is our plasmid. Plasmids carry DNA. Any gene of interest is going to have to be cut twice, once at the beginning and once at the end of the strand. Here's the beginning, here's the end of the strand. And then as they get combined, you make your piece of recombinant DNA. Recombinant DNA is then just placed in a bacterial cell, and now the bacterial cell can do whatever was in that gene. So that's the process, transformation. Last bit I'll add to this is that remember that recombinant DNA is created through cutting DNA. And in order to cut DNA, the cut area, which is done by restriction enzymes, has to leave behind sticky ends, or ends where the gene can actually bind to the plasmid. What this allows the DNA to do is really stick together. So if I cut my plasmid and split this end going to the left and that end going to the right, it will kind of open up, just like this is showing you right here. And then as you notice, this gene of interest being cut out and inserted into the plasmid could be potentially difficult to line it up correctly. However, the bases here match up, AT, AT, TA, TA, and because they'll stick together, it's a lot easier to combine them. So that's the last piece we'll add to it. All right, so what can you do with this kind of technology? Well, you can create transgenic plants, even transgenic animals, okay? Organisms that contain new genes. Transgenic plants would be anything like resistance to pests. Maybe the plants produce their own pesticide. Maybe they grow faster, and that way they produce more food. Maybe they have an increased amount of nutrients that the consumer may need. There are a whole host of different ways you can modify a plant in order to make it transgenic. When it comes to transgenic animals, we can engineer bacteria, maybe even some protists, to digest petroleum as a waste product, and in that way help us clean up oil spills. Okay, we can also allow them to grow faster, maybe be stronger, so we can harvest more meat or farm them out faster. So there are many different ways to modify animals and plants using this process, and that way they're transgenic. Here are two examples. We've got glowfish, which you buy these at Petco. They are modified zebrafish, and the zebrafish have a gene in them that allows them to grow, just for fun, really. In here, we've got a particular plant that has been engineered to grow when it needs water, interestingly enough. That's very advantageous for the farmer to know exactly when it needs water. But these are both cases of transgenic organisms. All right, so next we're going to get into the process of gel electrophoresis. What that was the creation of a DNA fingerprint. DNA fingerprints are basically extracted DNA from an individual person or species or organism, and through using, okay, so in here you use restriction enzymes to cut up the DNA, and in cutting the DNA you create individual bands that are unique. And by doing that, you can compare one person's DNA fingerprint to another's, and you end up, most of the time, seeing a lot of differences. Okay? What you are cutting is the human genome. Okay? There are 20,000 around genes that make up the genome, and you only have to cut the genome in a few different spots to start seeing differences between two individuals. The way you analyze those differences by using a process called electrophoresis, which is a way of taking the fragments, okay, taking the fragments of different sizes, putting them in a gel, electrifying it, and then afterwards, the largest segments move the slowest, and the shortest segments move the fastest down your gel, which is being analyzed. So electrophoresis is just a way of separating out different sized pieces of DNA so you can see the differences between them. Here would be one very extreme example, and here we have at least 200 samples in here that are being analyzed and compared to each other. This would most likely not be of individuals to see who's related to who. This would most likely be to see what genes of a species are related to other species. So this is a very large DNA fingerprint gel. You can see a lot of different samples in this one. In this example, we've got two, I believe, mice being compared with each other to see what genes show up in each individual mice. On the left, it's important to note, too, that sometimes you see gels here with what are called standards. You dealt with this in your baby lab. And a standard just tells you how big each segment is. So this top line here is 1,000 base pairs of long. Next one down was 600. That line's up there. 300 was just below that band. 200, and then 100. So it gives you a way of measuring how big the samples are in the gel, because the standard is a known sized piece of DNA. All right, so what is beneficial about this technology? Why is it good to have? Well, somebody with diabetes who has type 1 diabetes could potentially be helped because we can engineer bacteria to make insulin for them, okay? Or maybe even somebody who's short, we could engineer them to receive human growth hormone, which is just another protein from bacteria through transformation, okay? The creation of transgenic organisms could give us cabbage that are not destroyed by caterpillars, or maybe tomatoes that can, survive harsh winters, or apples that have higher nutrients in them. So all of these are benefits to using genetically modified organisms. Now, are there downsides? Of course. Okay, there are a few downsides. One being that we don't really understand all of the unintended consequences that could be brought on by using these. But they still do have some benefits. So a genetically modified organism, again, to just wrap this up, is just an organism that has been altered genetically. All right, finally, we're going to talk about stem cells and cloning. When it comes to stem cells, there are a few different kinds, but just be aware that a stem cell has the flexibility to change into many different kinds of cells. There are a few different types of stem cells, but for the most part, they can change into different kinds of cells. The most abundant, or not abundant, the most versatile stem cell would be called an embryonic stem cell, and they can be harvested from fertilized eggs and turned into any type of tissue you may need for a human being. So they are very useful, okay? In order to clone an organism, you would need some kind of cell like an embryonic stem cell, because it would have all of the DNA activated that would need to be activated to create a new organism. In order to do reproductive cloning like that, you need to first take the egg of one cell, the egg out of one organism, and empty out the nucleus, so it's empty. The next phase you'd have to go through is by taking any cell from the original donor animal and removing that nucleus, but you're not going to remove it and get rid of it, you're going to put it into the egg cell. Remember, now that that has occurred, this egg is diploid, which means it has all the DNA necessary to be an organism, and now that, when stimulated, will start to grow into an embryo, and hopefully, once it's implanted into a mother, grow into being a clone of the original donor, okay? This process you read about in your book was hard to do, but now that we're getting better technology through the use of stem cell and iPS cell technology, it's getting a little easier to do. Okay, Dolly here is the first cloned animal. It's a sheep, but we've cloned horses and bulls and cats and pigs and a whole bunch of organisms. So there's a lot of different cloned animals out there. Another way you could do this or use cloning to your advantage would be therapeutically. If you were to take the cells of your own body out or take a sample of them and remove the nucleus, place that nucleus into an egg and have it grow into an embryonic stem cell, you could then remove those stem cells from that cell and create organs that would match what you need, or tissues that would match what you need. Obviously, though, and many of you may have heard of this, the ethical issues surrounding this are vast. People are sometimes uncomfortable talking about using embryonic stem cells because it does involve terminating a fertilized egg in order to take the embryonic stem cells out of that cell. Okay. The last type of stem cell here would be the use of adult stem cells. And adult stem cells are multipotent, which means they can make a lot of different kinds of cells, but usually only in one category. And here we've got bone marrow that's producing a lot of different blood cells. Some of them they're not all red blood cells, there are other kinds of blood cells. And it's making those, but it can't really make anything else like brain tissue or muscle tissue. So adult stem cells can be used to make different categories of cells but not anything you want. Finally, when we look at the controversy surrounding stem cells and the different types of ways you could use them, always got to remember that there's a special type of stem cell out there now that's currently being used a lot more often than the other ones, called an iPS cell. And an iPS cell is a cell that has been differentiated, so it could just come from your cheek, could be any kind of cell on your body, but it has been re-engineered into an embryonic state without having to actually produce an embryo. No reason to terminate fertilized eggs, no controversy about using these. They are a lot safer, a lot easier to obtain, and people don't have a problem using them. So iPS cells are a great way to do the therapeutic cloning or the reproductive cloning that you're thinking of doing, and not having to go down such a controversial route to get that done. So that'll be our review here for genetic technology. If you have any questions, we'll go over the review guide you'll get during this period tomorrow during our Wednesday class. So have a good day, and I will talk to you tomorrow.